students. Today we will see something about the DNA, that is the genetic material that is there in our cells. Okay, so we will see how we will isolate DNA from the things that are available in our daily household, and even in our kitchens we have certain things. So from that we will just see how we can isolate and visualize DNA. Okay, so we are taking an edible fruit. Any fruit can be used for this purpose. So here we are taking a fleshy fruit, which is banana, and the first thing that we will do is we will try to crush the fleshy part of the banana, and for that I am using a Ziploc cover. So inside that we will first manually or we will simply crush the fleshy part of the fruit, and now I am going to take I will add some good amount of the common salt which we use in our daily life. So here it is the table salt that I am using. So you can add some 1 teaspoon of table salt, and we are going to use the same process of mixing it. And the Ziploc we are using just to avoid the mess that will be created if you do the same in an open container. So here we are going to mix this salt with the fleshy part of the fruit. The salt does the function of breaking the cell walls. Okay, so DNA, which is the main component for heredity, and it is the important component of every living organism, and it is responsible for carrying out the information from one generation to other, and it encodes all functional proteins, isn't it? So it will be tightly packed inside, and it will be very much securely packed inside the cell wall along with some complex of proteins. So we have to first denature all that, and what I just added is the hand wash solution. So this hand wash again does the function of breaking of proteins. Okay, so whatever protein is we can see in association with the DNA molecules will now be separated and denatured from the DNA, so that the DNA will now come into the solution in a naked form. Okay, so now this intense mixture of the common table salt, the fruit from which we are going to isolate the DNA, and the detergent or the hand wash solution, you can even use dishwash solutions. So that I am going to filter, so that this filtration is done mainly to separate the cellular part or the collective debris, and you are going to concentrate only on the the liquid part which may contain the DNA. Okay, so here we are going to collect the filtrate. So in laboratory conditions this will be done using a centrifuge, but here as we are going to exploit all the household equipments and materials, we are just filtering it with a normal tea filter. Okay, so here we are having this filtrate. Now you can see the filtrate is collected, and this filtrate now you can transfer it into a filter it into a narrow tube, okay, or a narrow vessel like a test tube. Okay, so either you can take a long glass like structure in which you can now transfer this filtrate. So here I am just transferring the filtrate into a narrow container, and to this filtrate to this filtrate now I will add alcohol. Okay, so in our household nowadays, every household will be having a sanitizer, isn't it? So the sanitizer can also be used here. So this alcohol is added to this filtrate in a gradual manner, and you can now see two immiscible liquids, and in that you can see a cloudy appearance which is your DNA. So here now we have successfully isolated something that is invisible to naked eye, the principal component of heredity. This cloudy part is the DNA that is the basic component of heredity and which is the most important material which encodes for all functional proteins in our body. So thank you.